Well, hello. Thank you for coming, and thank you for those of you who are attending online. It's my pleasure to welcome my colleague, Dr. Barbara J. Clark, who is an associate professor in the Department of Biochemistry and Molecular Genetics. Barbara received her Bachelor of Arts in Zoology from Miami University of Ohio, and then completed a Ph.D. at UT Southwestern, where she worked with Michael Waterman on the structure and function of cytochrome P450 hydroxylase enzymes, and during that time she published 5 papers. She completed postdoctoral training in endocrinology with Doug Stocko at Texas Tech University, and there she cloned and named the first steroidogenic acute regulatory protein, STAR, of which there's now a family of, I think, 14 proteins based upon their C-terminal start domain, which binds cholesterol, sterols, phosphatidylcholine, and other fatty acids. And I'm sure that Dr. Clark will explain more about this today in her talk. She was recruited to the UofL School of Medicine in 1995 and was promoted and tenured in 2001. She established a productive research program and published 36 peer-reviewed research papers and 18 book chapters here at UofL. And she's established productive collaborations with Eleanor Lederer and Matt Cave, and contributed substantively to their research efforts in the training of their graduate students in diabetic kidney disease and NAFLD. And of course, we're long-term collaborators with shared interests in various aspects of steroid hormone biology and their receptors. In her lab, she has trained graduate students and postdocs and medical students, and has continued to serve on numerous PhD dissertation committees because of her insightful approach to identifying key questions in current research problems and her compassion for students and graduate student education. In addition to her research, Barbara has been active in graduate and medical education, not only teaching the first and second year medical students in their curricula, and serving as course director for graduate courses in molecular biology, molecular toxicology, and BMG literature review, but as director of graduate studies in BMG from 2012 to 2018. From 2018 until just this summer, Dr. Clark was acting associate dean for the graduate school. Her service to UofL at the institutional, school, and department level is too vast for me to even begin to list. She has served on a variety of NIH, DOD, and American Heart Association grant review panels, including as chair. She has served on editorial boards for various endocrine journals and is currently deputy editor for the Journal of Endocrinology and the Journal of Molecular Endocrinology. So thank you, Barbara, for speaking with us today.
Well, first, thank you so much for that very nice introduction. I don't know if that makes me feel old or, but it's been an amazing time here at UofL, and that's mainly because of collaborators like Carrie had mentioned herself, and Dr. Cave and Eleanor Lederer and many others. So today I'm here to talk with you about my favorite protein, which is not in this title, but it's STAR D5, which is part of the START family protein, which I'll get into, and how it relates to endoplasmic reticulum stress, and looking at lipid metabolism dysregulation and non-alcoholic fatty liver disease. There we go. I have nothing to disclose. So just as a quick overview, we'll look at cholesterol and triacylglycerol metabolism in NAFLD, and then look at the links with endoplasmic reticulum stress and the unfolded protein response as they impact on lipid synthesis or dysregulation in disease states. I will introduce the START protein family member, and we'll focus on STAR-D5. And I pose this whether STAR-D5 is an anti-apoptotic factor in the ER stress response. But I do have to emphasize that this is a hypothesis. So hopefully by the end of this talk that I can get some, to give me feedback to see whether you think this is a valid claim. So to begin with this audience, I don't have to spend too much time on looking at non-alcoholic fatty liver disease and its progression, but just to get us started all on the same page, we look at the ability, of the liver, that if there is an accumulation of fat in the liver, which is observed with these fatty lipid droplets, that is the simple steatosis that is associated with non-alcoholic fatty liver disease. And basically, NAFLD is thought to be the liver manifestation of metabolic syndrome, which is basically a result of insulin resistance. And so obesity is linked to insulin resistance. So there is an association between obesity, insulin resistance, and fatty liver disease. Now, this is a reversible process. Diet can reverse the process back to the healthy liver. But if you have a second hit, what's shown here, it could be genetics, that will develop that into non-alcoholic steatohepatitis. And this is where we have infiltration of macrophages, inflammation, and sometimes cell death associated with this disease. And I want to look at what is important about the progression of this disease, because there's a lot of work trying to understand the movement, what shifts the balance in patients when they move from NAFLD into NASH disease. And some of the current ideas out there include the second hit hypothesis. So if there's a predisposition, genetics, as well as environmental factors, you can result in non-alcoholic steatohepatitis. But also, it could be, when I say environmental this way, it could be environmental chemicals plus genetics together. And this has been coined toxicant-associated steatohepatitis by Dr. Matt Cave in the Department of Medicine. So there are many, avenues really to get to. You can look at the liver disease, you can look at the fatty liver or NASH disease, but the etiology of the disease can be quite varied. So one of the things that we are going to talk about today is really looking at insulin resistance and how it impacts upon lipid metabolism and how that leads to non-alcoholic fatty liver disease. And this is a figure that we saw earlier this month, looking at this review from Lumba. And this is basically showing that if you have a Western diet where you have increase in carbohydrate as well as increase in fat intake, all this the carbohydrate will spark for increase in insulin secretion from the pancreas. And in the normal physiology, I would say that this increase in insulin signaling would signal for uptake of glucose into the glucose responsive tissues and its utilization. What happens in an insulin resistant state is that the insulin receptor signaling is blunted. And so in this case, the dysregulation in lipid metabolism that may occur is that you will have the increase in de novo lipogenesis, which means you're taking those carbohydrates and they can be, the acetyl-CoA can be converted into new fatty acids, and that can be those fatty acids are integrated into glycerol backbone to increase triacylglycerol levels in the liver. Now, while that is normal, what is supposed to happen is that those triacylglycerols will be packaged into VLDL, sent out into circulation, where they'll be taken up by the adipose tissue for storage. And that's where we expect to see our fat stored. But what is depicted here, we have lipid droplets that are accumulating, because in an insulin-resistant state in the hepatocytes, we have decreased in the VLDL production. We have an increase in lipogenesis and triacylglycerol synthesis, and these are being packaged into these lipid droplets so they don't become toxic. That's really as a protective mechanism. We also, if we look at the adipose tissue in an insulin-resistant state, we have a decrease in uptake of the fats from the circulating chylomicrons. So those chylomicron remnants with the triacylglycerols are coming back to the liver and adding to this increase in the triacylglycerol content of the hepatocyte. So that is a dysregulation resulting from insulin resistance, why we have fatty liver and accumulation of those lipid droplets in NAFLD. And this shows, this is a different view of the same concept. Again, this is showing where we have the factors that impact on obesity, that can increase, serum free fatty acid levels. We have the adipocyte dysfunction and adipokines that also contribute to the insulin resistance, where in the liver we have increase in lipogenesis, decrease in fatty acid oxidation. So we have an increase in those free fatty acids. And that is really one thing that's considered the lipotoxicity. If those have elevated free fatty acids, they aren't stored as triacylglycerols or in the lipid droplets. They can have an impact on cellular membrane integrity. And they also, this lipotoxicity can promote endoplasmic reticulum stress and mitochondrial dysfunction. So here, you can think two things that can happen. You have increase in reactive oxygen species that occur due to this increase in lipotoxicity and an increase in the endoplasmic reticulum stress. That will signal for an unfolded protein response. And together, these two signaling pathways, well, it's more than two, but these two signal for increase in inflammation, which would be associated with the development of steatohepatitis from simple fatty liver disease, and can signal for apoptosis. And what I can't discuss and will not be discussing today is a really hot topic, an interesting topic on how the gut microbiome can impact upon the lipid dysregulation in the hepatocyte through the inflammatory cytokines as is shown here in their signaling. But as I will focus on is looking at ER stress and how this pathways downstream, after ER stress is activated, how that impacts inflammation within the liver. So when we're looking at endoplasmic reticulum stress, everything that's signaling for it is termed the unfolded protein response, or the UPR. And this is just to take us through the ER stress and normal conditions. That what is shown here, there are three major players. If we start with PERK, PERK is bound by another protein, GRIP, glucose-regulated peptide 78. And when there's a presence of unfolded proteins, because one of the jobs of the ER is to promote protein folding. So unfolded proteins is a signal for a disorder within the ER. So this unfolded protein response is initiated. And that first step is dissociation of the GRIP protein from PERK. PERK homodimerizes and autophosphorylates itself, and one of the downstream targets is elongation initiation factor 2 alpha, which is involved in protein translation. And it typically will suppress protein translation within the cell, as this is an adaptive response, with the exception of activating transcription factor 4, or ATF4. This will increase the translation of this factor, which will then translocate to the nucleus, and you will have downstream signaling of target genes to increase stress response. IRE1, like PERK, is bound by BIP. Once BIP is the second name that is called for GRIP78. So once GRIP78 dissociates, you have homodimerization, autophosphorylation, and one of the major downstream activators for IRE1 is splicing of this XBOX protein factor, XBB1S, that generates a transcription factor that enters into the nucleus. And again, its target genes are going to be involved in synthesizing ER chaperone proteins, meaning these are proteins that will help refolding of proteins as a response, or activating ER-associated degradation. So together, the activation of PERK and IRE1 signaling through ER stress response will increase target genes that will help fold proteins and bring back ER to its homeostasis. A third member of the ER stress response is ATF6, and with loss of grip binding, you have ATF6 will move to the Golgi, be processed. And again, it's a transcription factor that will move and increase target gene expression. So what is also shown in this slide, and that we're going to look at, is the impact or the interconnection between inflammation and metabolism and ER stress. Some of the target genes not shown here that are not necessarily directly involved in recovery of the ER, I would just say, for an increasing protein folding. But you have direct targets within the lipogenic pathway, so you can have increase in lipid synthesis through PERK and IRE1 signaling pathways, and whereas ATF6 can decrease lipogenesis and gluconeogenesis through these pathways. If we look at inflammation, you can see that these signaling pathways can lead to activation of JNK and NF-kappa beta signaling pathways, which are involved in inflammation. And if we look at metabolism here, these are some of the regulators that are involved in lipid metabolism or cholesterol metabolism. So what we are observing is that you have activation of the unfolded protein response pathways that can also activate inflammatory pathways. They can also activate cell death pathways. And one major pathway for that is through the PERK increase in ATF4, which increases CHOP, which is a member of the cat enhancer binding protein family that will mediate apoptosis. And you have feedback regulation here, though, where you can see that you have cell death mediators that can impact signaling through the ER stress response. You have metabolic pathways. These are protein kinases that are associated with activating targets within the lipogenic pathways, can feedback and activate the ER stress response. And you have ER stress response proteins that can suppress or activate metabolism. So looking at ER stress response or the unfolded protein response has expanded over the last decade to more than just looking at maintaining homeostasis of the ER, but it has a major impact on metabolism. So I think the question then comes to the ER stress response or the unfolded protein response is certainly thought of to being an adaptive response. So here, if you have increase of these pathways, then you want to restore homeostasis. And that's either whether it's ER stress or the lipid homeostasis that you want to restore. But there is a switch, when it gets to too much damage within the cell, the signaling pathways can be activated to promote apoptosis. And I think, well, in my title, I have chronic insult, trying to dissociate that from an acute insult. But I think that's not probably the best terminology to use when you're thinking of these stress responses, but rather we think of this as decisions between cell survival or cell death. So the ER stress that mediates the unfolded protein response pathways that I just went over can signal for cell survival or for cell death. And this, again, just goes through some of the pathways. And what we can see, it's the same signaling pathways such as PERK can be activated, but if it doesn't increase CHOP activation, we might be going down a cell survival pathway as opposed to a cell death pathway. Okay. So again, this is just another look at this reciprocal relationship between metabolism and the ER stress response. And we have, signals that can induce ER stress. We look at that can impact on insulin resistance. We can have a backward arrow that insulin resistance can increase ER stress. And that we can see here that typically we associate insulin resistance with the development of non-alcoholic fatty liver disease, or NAFLD. But here, ER stress mechanisms may have a direct impact through regulating lipid metabolism in the hepatocyte to contribute to fatty liver disease. So it's a vicious cycle. I could say you have dysregulation of lipid metabolism that can further aggravate the ER stress response, and that can then impact on the disease state. And as we know in the beginning, it doesn't just have to come from obesity-related factors or high-fat diet. Anything that can impact upon ER stress, whether it's environment, chemical, and I just put STAR-D5 in here as a question mark, can potentially go through this pathway to exacerbate fatty liver disease. And so I wanted to get some human data to show this audience, and so I looked and found there's a study out there, and they are looking at, because my question was when I first started looking at this was, anybody actually looked at NAFLD NASH patients and to see if there's any difference in the unfolded protein response within these patients. They've done transcriptome studies for lipid metabolism, but in this report, what they looked at is basically just showing they had the liver samples that were from this liver tissue and cell distribution. I don't know if this is a public biorepository that people can access, but when they looked at did an Affymac microarray analysis, so basically they're looking at RNA levels of expression of these targets that are within the UPR, whether it's ER stress, lipogenic targets, autophagy, and apoptotic pathways. What they observed, if they compared just normal healthy liver to fatty liver, or the normal to the NASH liver, what they found that there really wasn't much difference between the normal and the steatosis and NASH with the ER stress markers or lipogenic markers that they were following in terms of being upregulated, but they were certainly all downregulated in the NASH livers compared to the steatosis livers. So this is suggesting that you have a decrease in this ER stress response as you're moving from, decrease in the markers. I want to make clear, decrease in these markers as you move from the NAFLD to NASH, but you're having a potential increase in the apoptosis and autophagy markers as you move from upregulation as you move from NAFLD to NASH. But they aren't really, the problem is I make that statement, but they aren't really comparing NAFLD to NASH. They're comparing it all to the normal tissue. So we really can't say anything much about the progression of the disease from this. Okay, now we're going to switch gears here a little bit, and I'm going to introduce you to the START protein family member. So the START domain protein family is named for the STAR, or stands for star-related lipid transfer domain, or START. And this is based off the structure of the steroidogenic acute regulatory protein, or STAR. That when we put this sequence into the database, back in 1995, there was no known hit that it could come. So at that point, it was novel. Well, very quickly, people that had greater skill in looking at bioinformatic data back then pulled out a family member based on sequence similarity, primary sequence similarity to this to the star protein, and there's highly conserved sequence similarity, but what is mostly conserved is a three-dimensional structure, which is shown here. And what is common in all the family members that has a start domain that has a hydrophobic cleft where these lipids can bind into this pocket. And the major lipid, as Dr. Kling indicated going into it, are cholesterol, and as shown here in the different family members, it depends on the cholesterol, sterols, which are oxysterols, phosphatidylcholine, and potentially fatty acids. So this is a mammalian start domain protein family, and again, what's cartooned here, they all share this conserved three-dimensional structure at the C-terminus that can bind lipids. And the family member that the subfamily member that we are going to be looking at is within the STARD4. And what is unique about this family member is that these are really soluble sterol transporters. Now, when we think of cholesterol transport, or I should say when I think of cholesterol transport, I usually am thinking of lipoprotein transport, between cell types. But within the cell, cholesterol needs to be distributed from the membranes. It's synthesized in the ER, but the ER has very low levels of cholesterol, so you have to have cholesterol moved from the ER to the plasma membrane. And I would say one of the ideas for the STARD4 family members, which includes STARD4 and STARD5, which I'm going to talk about, is that this is an intracellular movement of cholesterol. And in STARD5 in particular, there have been studies that have shown that it can move cholesterol from the endoplasmic reticulum to the plasma membrane and have increased efflux to the circulating lipoprotein HDL. And those studies really were, there's a group from Virginia Commonwealth University that's generated a STARD5 knockout mouse, and they isolated macrophages from those mice and were able to show that those mice had less plasma membrane cholesterol and less ability to efflux cholesterol to the circulating lipoprotein. I say that because really there's very little known about the biological function of STARD5. But the fact that it is regulated by, studies have shown that it's regulated by ER stress, and when we look at the tissue distribution, it has a very broad tissue distribution. This is from the Human Protein Atlas. But in the literature, the major cell types or high levels of expression where it has been found, then the liver, the Kupffer cells, and the macrophages. So these are the three cell types that are very intimately involved in liver disease, NAFLD, and the progression to NASH. And looking at immunohistochemistry analysis, we've also looked at STARD5 expression in the small intestine. This is a rat where you see it's at the basolateral membrane. And in the mouse kidney, we observed that it's expressed mainly in the proximal tubule cells, again, also highly localized to the basolateral membrane. So this is supporting that STARD5 at least is localized at a membrane site for cholesterol efflux. So ongoing work over the last couple of years in my lab has been really to define the biological function of STARD5. And to do that, we've generated knockout a mouse model as well. We use CRISPR-Cas9 editing, and not to go into the details of how we did this, but it was in a CD1 implanted into CD1 embryos, so we had to back cross the founder line, now 7 generations, to get it into a C57 Black 6 background. And the reason for doing that is this strain of mice is commonly used in studies for generating mouse models of NAFLD, because it is susceptible to high-fat diet-induced NAFLD and NASH. So this is a Western blot analysis that we're just validating that we have our knockout, where we see that expression on our knockout lines, both male and female, that we don't detect, or the very low levels expression of STAR-D5 protein. So we can use these mice, and I ask the question, is cholesterol lipid metabolism altered in the mouse model? Now, I went quickly through the STAR family protein members, and so there is potential for redundancy of function in there. So going into this study, we didn't think that there would be a difference between the wild type and the knockout mice unless we had a stressor. So the stressor that we use is tunicamycin. Tunicamycin initiates endoplasmic reticulum stress. It initiates unfolded proteins, so it will initiate the unfolded protein response. And this is used commonly to treat animals to generate mice that will have fatty liver or steatohepatitis. So we took our wild type and knockout mice and we treat them with a vehicle control or with tunicamycin, and after 1 and 4 days we harvested the liver tissue as well as kidney and ileum. I'm only going to show data for the liver tissue. And then did RT-qPCR to look at key regulatory genes in cholesterol and fatty acid metabolism. Now we're looking at this side, these are the major targets that we looked at. For cholesterol metabolism, we have transcription factors, the nuclear receptor LXR that's involved in regulating downstream genes for cholesterol synthesis, as well as the SREBB2. And we also looked at the rate-limiting enzyme in cholesterol biosynthesis, HMG-CoA reductase. For lipogenesis, we looked at the key transcription factor SREBP1C that is important for increasing enzymes within the fatty acid biosynthesis pathway, acetyl-CoA carboxylase and fatty acid synthase. And we didn't look directly at these genes, but we did look at steryl-CoA desaturase 1, which takes unsaturated fatty acids and makes a monounsaturated fatty acid. Cholesterol is also metabolized and converted to bile acids, and that is regulated through the nuclear receptor FXR, downstream short heterodimer protein SHP, that regulates the expression of the rate-limiting enzyme in the classical pathway for bile acid synthesis, which is CYP7 alpha hydroxylase. So we selected these key genes, one, because they are at the upstream controlling biosynthetic pathways for cholesterol and fatty acid synthesis. And 2, these are the genes you're going to see when you look at the literature for people that are looking at changes in fatty acid and cholesterol synthesis. Currently, the loss of STAR-D5, so the D5 knockout mice, did not affect their body weight or the liver to body weight ratio. And I will say tunicamycin treatment, the mice do lose weight with that type of treatment, and there's no difference in the weight loss between the genotypes either. So we wanted to make sure we had an ER stress response in these animals that were treated with tunicamycin. And if we look here at this is a Western blot again, looking at the protein expression for STAR-D5 and for GRIP78. If we look first at the GRIP78, we clearly see that there is an increase in the GRIP78 expression with tunicamycin expression, both in the wild type. This is a label, should be over here as well. These are the wild type animals, and these are in the knockout animals. And if you look at the quantified data, we can see that there's no difference in expression of GRIP78 between the induction by tunicamycin between the wild type and knockout or the basal levels. And we see the same thing when we look at the RNA levels by RT-qPCR, that we see no difference in basal levels or tunicamycin-induced. So this would suggest that in the absence of STAR-D5, the ER stress response is similar, as measured by this one target for the UPR. If we look at STAR-D5 protein levels, we see a modest but significant increase in response to tunicamycin treatment. This is quantified for the data shown here, and when we took more samples, we can see that increase. And clearly with the knockout animals, we see no expression of STAR-D5, which is what we expected. So these data show us that our animals are responsive to tunicamycin, the ER stress response has been induced, and that there's a modest effect on STAR-D5 expression in the wild type animals. So those data were from one-day treatment, and the RNA and protein levels are correlated. So here we're looking at the RNA levels for GRIP78 and STAR-D5, and this is in our experiment where we treated the animals for 1 day and 4 days. And again, you can see here that with the GRIP78 response, we see an increase in the both the wild type and the knockout animals to a similar level. And we don't see that, at the STAR-D5 at the mRNA levels, we're not seeing a change in STAR-D5 in the wild type. And as we expect, we don't see any effect in the knockout animals. So these data are to show you that we are sustaining that ER stress response, at least in the form of induced GRIP78 expression over our 4-day treatment in the animals. So now when we looked at select pathways and the impact of the knockout of STAR-D5, here we're looking in all the black indicate the wild type animals, and the knockout animals are shown in red. If we compare the circle, this is for the bile acid pathways, looking at FXR, SHIP, and CYP7A1. If you just look, at the black, compare the circles, the black to the red, in each of these cases, there is no difference between the wild type and the knockout animals for expression of these target genes. So in the absence of an ER stressor, there's no difference. And when we add an ER stressor, we see no difference for 1 day, which are the boxes, or the squares, and the triangles show the 4-day. We see no difference for FXR. We see no significant difference for SHIP. But with CYP7A, we see with tunicamycin treatment that we have a significant decrease in both the wild type and the knockout animals at 1 and 4 days, but there's no difference between the genotypes. And that's what's just shown over here, that we're seeing a tunicamycin effect only on the CYP7A. Now, if we look at cholesterol pathways in the STAR-D5 mice, with what we observe, again, you look at each of the circles and compare the black to the red for LXR, SREBF2, which encodes for SREBP2, HMG-CoA reductase, and ABCA1. Again, each of these, if you look at the circles, there's no difference between the wild type and the STAR-D5 knockout mice. And with the 1-day tunicamycin treatment, we begin to see a decrease in HMG-CoA reductase in these animals, but we see the same effect with the STAR-D5 knockout mice. So we can say there's a tunicamycin effect on HMG-CoA reductase after 1 day. But then when we look at the 4-day effect, what we're observing is that we're seeing a decrease in LXR in the knockout mice compared to the wild mice, a decrease in HMG-CoA reductase in the knockout mice compared to the wild type mice, and a decrease in ABCA1 in the knockout compared to the wild type. So this will be suggesting that we have a genotype effect where loss of STAR-D5 does decrease LXR, HMG-CoA reductase, and ABCA1 expression. Okay. Now, looking at this, I'll just point out what you observe here is after 4-day treatment is that here we're seeing, I will say, a recovery potentially in these expression in the wild type mice of these targets, whereas we are maintaining this low level in the knockout mouse. Now finally, when we look at the lipogenesis pathway, we looked at 2 SREBF1, which encodes for SREBP1C, and SCD1. Here, again, there's no genotype, looking at the genotype difference and without a stressor. After tunicamycin treatment, we see a decrease in the expression of both genes, SREBF1 and SCD1. And at 1 day, it's similar between the wild type and knockout mouse. But at 4 days, we see that the SREBF1 maintains a lower level of expression, whereas the wild type mice has a recovery. But with SCD1, we see a further decrease in expression compared to the decrease with the wild type mice. And I will note, if you look at the y-axis, this is a significant change. We're looking at, this is over 100-fold difference between these changes in gene expression. So it is quite a striking difference here. So when we look at this in terms of the lipogenesis pathway, we see a tunicamycin effect that it impacts both wild type and D5 knockout mice. So we have a decrease in SREB1C and SCD1, the 2 that we looked at. But we also see with longer term treatment that is the decrease in SREBP1C and SCD1 is maintained in the knockout mice. So what does this mean? Well, if we look in the literature, we know that tunicamycin-induced ER stress in C57 black 6 mice have been shown to decrease some of these targets that we were looking at. So SREB1C, SCD1, HMG-CoA reductase, and CYP7A1 are just a couple that I have highlighted and looked at. But when we look at our STAR-D5 knockout mice, what we're observing is that in longer term treatment, we have an impact on the lipogenesis genes and the cholesterol metabolism genes that are not observed in the wild type mice. So if we think about this, bringing this together, that the ER stress pathways, their metabolism can impact ER stress, and ER stress can impact metabolism. And if we look at the unfolded protein response targets, the transcription factors that are mediating these different pathways, we know that ATF4 and XBP1 and ATF6 can directly regulate these targets in lipogenesis and cholesterol synthesis. So if you activate ER stress, you can have activation of the lipogenesis targets. If you activate ATF6, you can suppress some of these upstream regulators of lipogenesis. And if you activate CHOP, you can have an increase in apoptosis and suppress some of the signaling in the lipogenesis pathway. So one thing, this becomes very complex very quickly. So trying to find targets that you might want to target or clinically target, because you want it would be helpful to understand the impact that each of the, that the ER stress is playing on the metabolic pathways, as well as the UPR targets that are being activated and how they interact. Because here clearly we're seeing that we have ER stress pathways that we need to see what activation of these UPR targets are occurring, because we're seeing a decrease in the lipogenesis genes whereas some of the prediction would be to see an increase. But we have to take into consider the time frame, because again, are we looking at adaptive or are we looking at cell death responses? Are we looking at cell survival decisions versus cell death decisions? And I'm proposing that in the STAR-D5 mice that they may be more susceptible to the cell death decision making of these pathways, because in the STAR-D5 knockout mice, the impact on the lipid metabolism may be modulated. So that brings me to, does STAR-D5 serve as an anti-apoptotic factor? Because we saw when we knocked out STAR-D5, we had these results. I'm going to focus right here down on the lipogenesis pathway. Because if you have a decrease in the conversion of saturated fatty acids to unsaturated fatty acids, this can increase lipotoxicity and promote pro-apoptotic signaling. So you have, this is actually from a glioblastoma stem cell study, not disclosures, not NAFLD study. But in this study, they were looking at inhibitors of SCD1 and how it can promote cell death. And what is shown here is if you have under, I want to say, an adaptive ER stress response, when you have balance between saturated and unsaturated fatty acids, you have SREBP1 signaling, modifying the lipogenic gene expression. And if you have unsaturated fatty acids, that's a protective mechanism. But if you have loss of SCD1, there's going to be an imbalance, and this is going to shift to the junk signaling pathway and going through apoptosis. So it could be through this pathway that STAR-D5 may be playing an anti-apoptotic role, because in its absence, this is elevated. And several years ago, this is a collaboration that Dr. Kling and I worked on when we're looking at STAR-D5 and lung cancer, I would say. And there we found that STAR-D5 is overexpressed in several cell lines of lung adenocarcinoma cell lines compared to normal human bronchial epithelial cells. And this just shows one of the work that when we knocked out STAR-D5 through shRNA in this one cell line, 1792, we looked at doxorubicin-induced apoptosis, looking at caspase 3-7 activation, and we could see that we had an increase in apoptotic signaling in the absence of STAR-D5. So in the back of our minds, we've always been thinking that, how is STAR-D5 playing a role in apoptosis? So that is where this leaves me, and to leave you with this thought. In the question that we are writing a grant on to trying to see if STAR-D5 knockout mice are more susceptible to ER stress-mediated liver inflammation and apoptosis. And that comes to, if you think about in the hepatocyte, and in this case, using a high-fat diet or a diet that will induce fatty liver over a chemical-induced, pharmacological-induced tunicomycin that would only impact ER stress directly and not go through potentially the lipotoxic pathway to get to ER stress. That makes sense. Our preliminary studies have used tunicomycin, which is only going to go directly to ER stress. We want to ask the question in the normal pathophysiology of most patients with NAFLD that's going through insulin resistance and lipotoxicity. If these mice on a high-fat diet, you would expect them to develop this disease. And if STAR-D5 is present, you're going to have dysregulation of lipogenesis and cholesterol homeostasis. But in the presence of STAR-D5, you'll have more movement of cholesterol to the plasma membrane for efflux. And you can have some potential controls here on still mediating saturated fatty acid to unsaturated fatty acid biosynthesis. But in the absence of STAR-D5, if we have this suppressed and we have an increase in saturated fatty acids, this can feed back and further promote the lipotoxicity and ER stress. In the absence of STAR-D5, if we have less movement to the plasma membrane, we can have cholesterol in the ER that's going to promote further ER stress. And all this overactive stress response, you go through CHOP-mediated and junk-mediated apoptosis. So that is where we are now, the questions we are asking, and certainly, with this audience, very open to any clinical relevance or where in any area that you think that we should be focusing on, in terms to get where you think that movement of cholesterol within the cell and controlling saturated fatty acid to unsaturated fatty acid metabolism might be clinically relevant. So with that, a big thanks to Kellyanne Peel, who's a technician in Dr. Kling's lab, who's doing all the work. And this is an old slide, and I must acknowledge Vishnu Patel, who is a medical student who did a lot of the qPCR analysis this summer while he worked in my lab. Collaborators I've had on kidney disease was Dr. Lederer and Jalal Kundamari, who's now at Howard University. Ron Gregg and Tim Hoffman. Dr. Gregg's the chair of our department. He's also the one that generated the knockout mouse model for us. And again, Carrie Kling, Dr. Kling has been instrumental in helping me think about the project and support both with sharing her lab space with me as well as her technician and giving me great insight. And Alan Chang, who's a previous faculty member who's now working for Abbott on the diabetes, developing insulin monitors for diabetic patients. Also instrumental on the side for looking at using the knockout mouse model to look at questions to address lipid dysregulation. With that, I thank you for your attention, and comments are welcome. And I think there was...
Just stop sharing.
Stop sharing. Okay. And I have chat. Okay, there's a lot in the chat. Uh-oh. Okay. Yeah, I hope you did hear me. I had the thing that there was no audio receiving, but I guess I got that.
Okay. Should have been fixed.
Okay. Okay. So, does the ER stress result in signal peptide cleavage and glycosylation of membrane proteins? So, yes, I understand. Let me think. So you would have a accumulation of these proteins within the ER that promotes that stress. So that would impact upon the signal peptide sequence and cleavage that could occur and secretion of those proteins. So any protein that is transiting through the ER has the potential to be impacted in an ER stress response. And that's what I guess I should further say what the UPR is doing is some of those target genes will generate chaperone proteins that will come back and then help fold those proteins in the ER and reset homeostasis, so you will have signal peptide cleavage and folding of those proteins. If you can't move out of the ER and get into the Golgi, for glycosylation, that would impact glycosylation as well. And you're right, I didn't mention the mechanism of action of tunicamycin. So using tunicamycin to stress the ER is likely to have more effects than intended. What are your thoughts? Yes, so that's a great point, because this is a direct, harsh treatment within in the ER. So you're really having a response, and that's the one reason we did this is because it's a short-term treatment, and you can generate fatty liver, and this is probably not going again through the lipotoxicity through the fatty liver normal pathology for NAFLD, but it's asking the question, if does ER stress, and what was showing and supporting is data out there that if you have induced ER stress, you're going to impact lipid metabolism, and that can have its self cycle. So I think, yes, the more effects than intended. It'll be interesting if you have with the high-fat diet. A lot of the studies have been on the high-fat diet treatment of these mouse models, C57 mice, may show that there is an increase in the lipogenic pathways, because that is what is observed in patient samples and it's in other samples. So we need to address that. Can any other stressors be used instead of tunicamycin? I think I hit upon this. Yes, I think there's a methionine choline deficient diet, which is commonly used to induce NAFLD in mouse models, and that is also associated with increased ER stress. High-fat diet, again, also is associated with induced ER stress. And that's why moving forward, we want to look at those models over using the tunicamycin for the points that you raise. And it is a not pharmacological, it's a chemical insult to the cell.
What does insulin resistance do to our B cell? Does it have a direct effect for insulin?
That's a great question. We don't know what insulin resistance does to STAR-D5. So I've been trying to data mine if there's any expression profiles out there, if we can pull out STAR-D5. But I'm looking at NAFLD models, but it'd be nice to, I haven't seen that. I haven't uncovered anything yet.
And what about?
It's not that I haven't found, let's say I haven't found anything yet. So, okay.
And what happens to the adipose tissue?
In terms of STAR-D5, no one's looked at the START proteins. We saw in the human protein atlas that's expressed there, it's UofL, but no one has looked at adipose tissue for that. And then hepatocytes, it might be, and I'll say bias, my bias is I think it's more, which I didn't present here, the impact might be more in the macrophage than in the hepatocytes for STAR-D5 in particular, in terms of inflammatory.
Knockout, not liver specific.
No, it's a whole body knockout. Yeah. I think with the inflammation response, that could be due to macrophage recruitment, and we know STAR-D5 plays a role. It's expressed in macrophages and does the same in terms of cholesterol trafficking in macrophages.
So I would expect that based on your data that you would have decreased cholesterol in the plasma membrane of the knockout mice. Can you test them?
Yes, you can. You can isolate the. I guess you could isolate the hepatocytes. I mean, that's what the other group did, isolate the hepatocytes, and then they used fluorescent tagging of extracellular exposed cholesterol to detect plasma membrane cholesterol levels. So I think if I mean, we did a little bit of that early on where we would hard to isolate plasma membrane from ER membrane by differential centrifugation. So I think you need to have pretty sophisticated techniques to look at that cellular distribution. And most of that's done with fluorescent labeling of cholesterol and looking at its movement.
So it's not easy to just take a chunk of liver and do some sort of on it and see. Although maybe if you have decreased MUFA and increased palmitate.
Well, yeah, that's what is, that's what I'm interested in thinking. If we can look at the lipid profile and to see if there's any difference between the knockout.
So is there a way to overexpress RB expression?
Yeah, we probably do adenoviral.
Is there any other, just weight loss?
Well, so ER stress itself is supposed to induce STAR-D5. That's what has been reported in the literature, although there's not, I would say, solid data. It's supposed to have been at the RNA levels. We don't see it in our animals, which was a shock. I mean, based on my expectations of what has been reported, I expected to see an increase in D5 that parallels growth.
They use the fatty fat, insulin synthesis, like EPAR gamma.
Yeah. No, so I think the question of overexpressing STAR, because I think in the challenge here is two things that we have observed is that there's no difference in the knockout basally. So it's not essential for these metabolic pathways. But it's only when you have a stressor and potentially when you have chronic disease that you will then begin to notice an impact of STAR-D5. And that's with the 1 day versus a 4-day tunicamycin treatment. So now, even with the high fat diet, is that going to be sufficient to see a distinction between the knockout mice and the wild type mice? We may need another insult. If this is harshly hitting, I think, idea of tunicamycin is really impacting the ER. It might be you need very severe ER dysregulation to notice an impact to STAR-D5. And if it can protect from that long-term disruption, that would be important to find out. Okay. Any other questions online? Okay. Thank you.
Thank you very much.
I just push stop recording. That's all I do is just press stop recording.
Yeah.